Did you all get the email for the lab quiz study guide? So on Monday, we can answer any lingering questions you might have about the lab quiz. There will be no reading quiz for Monday. I'm going to give you an exam Friday and then make you do a reading quiz for Monday. So really your next thing would be to do some studying for the upcoming lab quiz. We'll talk about that. And then in addition to taking the lab quiz, next week in lab you will be working on your data. You've got to average your data that you've collected this week. And then you can also start working on your PowerPoint presentation and your group presentation. And there's no pre-lab quiz for this coming lab, right? Okay. So, does anybody have any questions you really need to get answered about the exam? Nothing? Well, you want to see how you do you want some questions? I don't know if the clickers are working, to be honest with you. We'll have you try responding to this one, and then we'll see if results come up. It seems to me that I remember kind of hiding the poll response question at one point in time. But let's start out. See what you think about the first one. Which statement correctly describes catabolic pathways? So I'll give you a couple of seconds to put in a response, and then we'll see if we get any tally of responses. If not, we can just go through these. Okay, looks like we're getting some response here. Okay. So the majority of the students did get the question correctly. They do release energy as they degrade polymers to monomers. Just because a reaction is catabolic, does that mean that it doesn't require an enzyme? Not necessarily. The breakdown of glucose into carbon dioxide and water is a catabolic reaction. But it occurs at a very slow rate. And so remember that concept that enzymes increase the rate of reaction even if the reaction also is spontaneous. Okay, again, the majority of the class is correct. Which law of thermodynamics says that living organisms can convert energy from one form to another? The first one. There's like two laws of thermodynamics that we talked about. The first law of thermodynamics. Living organisms, to be alive, must use energy to maintain their organization. And maintaining their organization, which requires energy, decreases the entropy, okay? Remember that idea of entropy being a measure of disorganization. So highly organized cells are states of low entropy. Okay, we're on a roll. Again, correct answer. Proceeds with the net release of free energy. You can see that there's some themes here already if these questions are kind of typical, and there's some just understanding of basic terminology, almost definitions of exergonic, endergonic, catabolic, anabolic, in terms of entropy. So to answer questions, you have to know what those terms mean and refer to. Systems at a high state of entropy are unstable. That's false. Okay? Remember, high states of entropy is high levels of disorganization. And what does the second law of thermodynamics say? Things are going to get disorganized. That's a natural tendency. Things tend to go from unstable states to stable states. So disorganization is the most stable state. Organization, or low states of entropy, is the most unstable state. Cells are inherently unstable. Mistake on this slide. A cell is a system with a low state of entropy. It's organized. And therefore it is an unstable state. So it should be 1 and 3, not 2 and 3. That is a true statement. Okay? If cells are inherently unstable, the second law of thermodynamics says everything tries to get stable. So cells should increase their entropy. Their natural tendency is to die. How do they counteract that? They expend energy to maintain that state of instability, that organization. Cells are open systems, right? Do cells increase the entropy of their environment? Yes, they do. Okay. That's how you get around what seems to be that contradiction of the second law of thermodynamics. If a particular part of the system is going to decrease its entropy, there's got to be a corresponding increase in entropy in the environment. So cells live basically at the expense of their environment. They maintain their organization by causing a greater level of disorganization in their surroundings. Cells don't only have exergonic metabolic reactions. We saw that there were some endergonic metabolic reactions in glycolysis. Some of those reactions require the input of ATP. Do cells drive exergonic reactions by coupling them to endergonic? No. They drive endergonic reactions by coupling them to exergonic reactions, like the hydrolysis of ATP. So in that particular question, make sure you read carefully. Okay? Back on the right track again. ATP as hydrolysis is spontaneous. It's an unstable molecule. It is an exergonic reaction. It's an energy-releasing reaction, and therefore has a negative delta G. What's going on? It's releasing more energy from bond formation than is being used up in bond splitting. In ATP hydrolysis, you have to chop off that terminal or third phosphate group But that doesn't require much energy because that bond is highly unstable. Water molecules also have to break bonds, but water spontaneously dissociates, and so that doesn't take much energy. But when we reform bonds, as ADP and phosphate are formed, more energy is released from that bond formation than is used to break the bonds, and that's why it's energy releasing overall. Okay, so we got free energy of reactants, free energy of products. Spontaneous or not? It's spontaneous, right? The reaction is downhill. The reaction proceeds with a decrease in free energy. So the only one that is correct there is the reaction is exergonic. All over the place on this one. Okay? Remember when we broke down reactions into two basic forms? We said catabolic and anabolic. Okay? Catabolic tend to be those exergonic processes. Anabolic tend to be those endergonic processes. Catabolic reactions are oftentimes oxidation reactions where substrates have hydrogens removed. The anabolic ones, which are typically endergonic, tend to be reduction reactions where hydrogens are added on to the molecule. Endergonic reactions tend to be reduction reactions, not oxidations. Endergonic are not spontaneous, they're uphill. They're not associated with increasing states of entropy. They take something that is less organized and make more organized molecules. Simple molecules into more complex. So they're associated with decreasing states of entropy. So none of those above statements was correct. Okay, good. So what represents the energy available to do work? That's what delta G represents, right? What's delta G when a reaction reaches equilibrium? 0. So that reaction can no longer produce energy to do cellular work. That's why the cell organizes its metabolism into these metabolic pathways. So that once something is produced, it is again then used in a subsequent reaction. That organization in the pathways helps to keep the reactions from reaching equilibrium and basically keeps metabolism running. Okay, the first question is, what is feedback inhibition? And feedback inhibition, remember, your end product of a pathway can go back and inhibit an enzyme earlier in the pathway, thus shutting down production of that end product. In order for that to happen, the inhibition has got to be reversible. If it's non-reversible, then things just shut down permanently. And then metabolism can't respond to changes in the end product. So those non-reversible inhibitors typically bind to the enzyme by strong covalent bonds. And once they do that, they basically denature the enzyme and everything stops for good. That's why those compounds like mercury and arsenic are poisons. They denature enzymes and kill cells. So the enzyme's got to be reversible. There are two types of reversible inhibition. There's competitive and non-competitive. And allosteric inhibition is a type of non-competitive inhibition. So these are all examples of reversible inhibition. So make sure you understand the difference between competitive, non-competitive, reversible, irreversible inhibition. Okay? And this is a real key. The fact that this oxidized NAD is present in limited amounts in the cell, that's why if it all becomes reduced and you run out of the oxidized form, things start stopping. Those dehydrogenase enzymes that require the oxidized form of the coenzyme, if they can't work, then glycolysis can't work, citric acid cycle can't work. Remember, reduction of molecules increases their free energy. If you add electrons or hydrogens to them, then later there's an opportunity to remove those molecules. And as you remove the electrons, you can potentially make some energies from something like an electron transport system. Cellular respiration, there's a lot of times and a lot of places where oxidized NAD becomes reduced. So where are those electrons coming from? They're coming from glucose or some other organic molecule that can be processed into either glucose or some of the intermediates of glycolysis or the citric acid cycle. So the source of the electrons are those food molecules. Where do the electrons end up going once they've been temporarily put onto reduced NAD? Ultimately, they're going to be transferred through electron transport to oxygen and make water. So water is the final resting place for those electrons. It's not the source of the electrons. So that's an important distinction. Anybody know the significance of vitamin B12? Remember that? What's relevant about vitamin B12? That is one of the molecules required by the cell to manufacture its coenzymes NAD. Recycling or reduced coenzymes during electron transport allows us to produce ATP by oxidative phosphorylation. Remember that substrate level phosphorylation doesn't require an electron transport chain. nor does it require any membranes. Activation energy, that hump that the molecules have to get over to reach that unstable transition state where their bonds are just ready to break, is the energy of activation barrier. And what do enzymes do? They lower that barrier and make it easier to get over that hump. Coenzyme. Do all enzymes require a partner coenzyme? No, only some do. What's the most common one that we've talked about in cellular respiration that requires a coenzyme? What class of enzymes? Remember the dehydrogenase enzymes, the one that remove hydrogens from substrates? So that hydrogen's got to have some place to go temporarily. It goes to the coenzyme. I don't know if you noticed, but on all of the multiple choice questions at the very beginning, I always ask you to choose the best answer. This is an answer, but not all reversible inhibition is feedback inhibition. This particular example talks about specifically the case of feedback inhibition. So you could argue reversible inhibition is a correct choice, but it is not the best choice. Feedback inhibition is. Loss of electron or hydrogen oxidation. Gain of electron reduced. Okay, so glycolysis 1, citric acid cycle 2, etc. Where is CO2 produced? Producing glycolysis? No. Producing electron transport? No. Only in the citric acid cycle. Important. When I say fermentation, what am I talking about? I'm talking about the pathway of glycolysis plus some additional step or steps, which is going to then recycle that reduced NAD produced during glycolysis. So don't confuse and equate glycolysis with fermentation. Okay, there are two pathways. Fermentation contains glycolysis, but it contains additional chemical reactions as well. Substrate loss level phosphorylation, citric acid cycle, and glycolysis both. correct there. Same answer again, correct? Glycolysis and citric acid cycle. Correct, electron transport chain. If I had fermentation up there as a choice, would you have included fermentation in that then? Yes, you would have. Okay. Is there any place in glycolysis that oxygen is a reactant? No. Is there any place in the citric acid cycle where oxygen is a reactant? No. The only place where oxygen is directly involved, it's one of the reactants, is in the very final step of electron transport as the acceptor for those electrons going through the electron transport system. So it's only in the electron transport system. Only one place where ATP is used. That is in glycolysis. Only one place in which reduced FAD is oxidized, electron transport chain. Only one place where oxidative phosphorylation occurs. The sequence where oxygen is directly involved, the electron transport system. Read your question carefully. How many ATP produced? It doesn't say how many net produced. 4 are produced, 2 are used up. But the answer to this particular question is 4. Some of you might argue that this should be last choice, the correct choice should be 36 to 38, and you're probably right. But of those particular choices, 38 would be your best choice. So those are all pretty straightforward kind of questions that you should be able to answer. Dr. Morton, as someone who is sitting in the back kind of observing all the time, any recommendations you would give them for exam preparation besides what I've told them?
Small groups, ask each other questions, go through processes. It always I think it's best when you can work with 2 or 3 other individuals and challenge each other. Studying by yourself, you're just not going to necessarily have a broad perspective on stuff.
Well, and when you study with other people too, the act of explaining something to someone else
Oh, yeah.
really means you understand it.
I can't tell you how much biology and chemistry I learned when I had to teach it.
Right, yeah, that's absolutely true. Going over the old exams is a good way to kind of do final preparation to see if you really do understand those particular concepts, okay? When you're doing multiple choice questions, I try to underline, negative descriptors like false and not and things like that, but be aware of those and read your questions carefully. I think it'll be helpful this time that we're only doing 2 chapters. I understand you had a chemistry test over just 1 chapter, is that correct? So a little bit less information, but the energetics material and respiration is something that tends to be confusing to students too. So.
After Friday, I would strongly recommend that you take the time to go over the study guide for the lab test. Study guide, if you can answer the questions on the study guide, the lab test will be quite easy. I wouldn't say that today because it won't be possible to do that tomorrow.
Right.
But on Monday morning, we will take questions if you have any on the lab material.
Yeah, and your old lab handouts will be very useful in terms of, and
A lot of what you will be challenged to do on the lab exam are the same types of things you're challenged to do on lab hand-ins. So if you use the hand-ins to go over what you did in a particular lab, you'll be well prepared.
any last minute questions? Well, study hard, but get some sleep as well. Don't underestimate the value of sleep before an exam. And if you want a format where you can do some small group work, go to the mentoring session tonight. Austin will be there to help groups. 8 o'clock.
Dr. Moore, just looking over one of the past exams, and there's a question that was like, what's the opposite reaction? It was something plus PI.
Oh, PI stands for inorganic phosphate.
Oh, okay.
Okay.
Yeah.
So typically when you have oxidative phosphorylation, you've got that inorganic phosphate getting stuck onto ATP to make ATP. And substrate level phosphorylation, the phosphate is part of another organic molecule. So then it's not referred to as PI, so PI stands for that inorganic phosphate.
Okay. Because like the answers was like E plus PI equals EPI and then C plus PI.
But did they give you some delta G's with them?
Yeah. Yeah. Have you?
Okay, what you need to do is you need to basically look and see if it's an endergonic reaction that's got a certain delta G value.
Because it was like
your exergonic has to be greater than that.
Okay.
A greater negative value than the positive value
Okay.
required
Because
for the
it
reaction.
went the other way, so it gave you a negative value, so then it has to be less than the positive, or like.
I'd have to look and see.
Because it was like -7 and then +5 was the one that was the answer, but there's another that had +9. So I didn't know how to.
If
choose.
you're around and you have the exam, drop by and I'll take a look
Okay.
at it and see.
Okay. I'll come back later.
Okay.
Was it extra credit or whatever online for tomorrow? Or what is it?
Oh, the optional points?
Yeah, optional points.
Yeah.
And, okay. Is it just like a crossword again, or
I forget what's on there. There's a couple crosswords.
There's crosswords on there.
you just turn in one of them, or?
You can turn in both of them.
Okay.
Yeah.
Just my phone.
So, how do you feel?
I think I'll be fine as long as I listen to the lecture from Tuesday and finish because I'm halfway done with this 7.
Have you checked to see if that's now posted?
Not yet. I was going to do that today.
Why don't you stop by Natalie's office? Oh, she's not there today, is she? I hope she had it posted.
I can check.
Check and see and let me know.
Okay.
If she didn't, it would still be on here, and I could just let you use the recorder.
Okay.